also been the National Postdoctoral Fellow, School of Life Sciences, Jawaharlal Nehru University, JNU, New Delhi, from April 17 to April 19. If I talk about his academic qualifications, so he has done his PhD in neurobiology and life sciences from Defense Institute of Physiology and Allied Sciences, molecular mechanism of hypobaric hypoxia-induced neurophysiological perturbations from New Delhi in November 2016. He has also been advancedly trained in the CAGA Advanced Neuroscience Training in Brain Homeostasis and Neurovascular Coupling from Bordeaux, France in June 19. Advanced Wellcome Trust course on System Biology and Functional Genomics in June 13 from UK. Molecular Neuroanatomy course from Okinawa Institute of Sciences and Technology from Okinawa, Japan in year 2011. Sir is also being a lifetime member of International Society of Cerebral Blood Flow and Metabolism and also a lifetime member of American Chemical Society. So over to you, sir.
Okay, thank you. I'll just share my screen first. Hello, very good morning and thank you for a very kind introduction. So today's session is on Western blot, and in this program I will try to be as concise as possible. So basically we have to understand that in order to understand any cellular function, we have to understand the protein's role, because the protein play a very important role in the cell. And if you want to understand how the expression of protein is changed, how in different condition the protein is changed, it's very important to understand whether it is present in a system or how the quantity of the protein is changed. So first seminal paper of Western blot was first published by Harry Tobin in 1979, where he first showed that electrophoretic transfer of protein from a polyacrylamide gel to nitrocellulose sheets. And this is the basics of any blotting. Actually, in any blotting system, the content of a gel is transferred into membrane. And in the case of Western blot, it is a protein. So Western blot is an effective and widely utilized immunoassay that confers selective protein expression analysis. And it exploits the property of antigen antibody binding in conferring the main principle of Western blots. So what are the main principle of Western blot? Actually, the Western blot is mainly to identify or quantitative protein. So to identify and quantitative protein, we have to first make sure that in a condition or in control condition or in experimental condition, we have to first equally load protein, or we have. And the second important principle of Western blot, that it separates the protein by molecular weight. So to understand this, first I want to make sure that this is a classical Western blot setup, where there is a tank where the gel is loaded and it's buffer. And in this electrophoretic setup, the proteins are separated on the basis of molecular weights. And the lighter molecular weights travels far, and the protein with higher molecular weight travels slowly. I will explain in details about each principle in subsequent slide. So we have to first appreciate that the Western blot technique precisely based on these four pillars. First, equal loading of protein. Second, separation of protein by molecular weight. Third is a transfer, electrophoretic transfer to a suitable membrane. And last, we have to probe this protein, and we have to probe this protein with a particular antibody. It's specific to the protein of interest. So first, I'm coming to the equal loading of protein. Equal loading of a protein is the first step of equal loading of protein is cell lysis. Whether it is a prokaryote cell, eukaryote cell, or a tissue, the first and foremost thing for a Western blot is the cell lysis. We have to get this protein out of a cell, and this protein extraction, depends upon the type of buffer that we use. Generally, we use RIPA buffer, and this RIPA buffer consists of NaCl, tris-HCl, and ionic and non-ionic detergent. The idea is to break the cell membrane and get the protein out. And in the process of a cell lysis, we also use protease and phosphatase inhibitor, so that we can maintain the structure of a phosphorylation status of a target protein. So if you see here, the kind of a protein that we get from a cell after the lysis is generally positive and negative charge, or have a disulfide charge. So it may be monomeric, it may be dimeric. So we will sort out this issue in subsequent slides, that once this protein is lysed after the cell lysis procedure, we next estimate the protein. And this is all to satisfy our first objective, to equally load the protein. So because the sample or the experimental samples may differ in the quantity of a protein, so we make sure that each well of an electrophoretic gel is loaded with a similar quantity of protein. So we adopt a methodology called Bradford assay. Here it is a calorimetric protein assay, where there is a dye called Comessi Brilliant BG250, which binds the protein and gives a color. So what we do is that we make a standard of this, and with a known concentration, we plot a graph. For example, if you know that, for example, we take a BSA and this Bradford assay, and we know 1 mg, 2 mg, 3 mg. With a known concentration, we plot a graph, and then we also do a Bradford assay of our protein, sample protein, and then we quantify the protein based on this regression analysis. And we generally load 20 to 30 microgram of total protein from a cell lysate or tissue homogenate that I explained earlier. And third step is the I'm just coming to the slide. This slide is first, is the sample preparation. So once we have a cell lysate and we quantify the protein, then we prepare the sample. And the sample consists of three things: protein extract, cell lysis buffer, and a lamellae buffer. And this protein extracts is generally gets normalized with the cell lysis buffer. And we have another lamellae buffer, which consists of a tris-HCl, glycerol, SDS, beta-mercaptoethanol, and bromophenol blue. And these all, the purpose of this lamellae buffer is two purposes. First, it's add density to the sample, so they do not, they drift properly in the gel. And second is the addition of this a negatively charged use of SDS and SDS with a negative. Just I explain here. The bromophenol blue is a non-reactive reagent that serves as a dye front for electrophoresis. And SDS is a potent anionic detergent that coats denatured protein with an equal anion to mass ratio. This mass protein charge, shape, and size characteristic and renders them solely to function of a molecular weight. And last is beta-mercaptoethanol is a reducing agent that acts on a disulfide bond. So if you see here, the disulfide bonds are first broken into monomeric form, monomeric form, and the protein is coated with equal charge to mass ratio. So finally, what we get is the entire protein, whether it's earlier after cell lysis positive or negative. Now after the treatment with sample buffer is all negative. So in the gel, they are separated on the basis of mass only, because the charge is already the same. And next is the separation of protein by molecular weight. And here, after the loading of a gel, we have to understand the constituent of a gel. So the gel consists of basically it is used in Western blot is SDS page. It was sodium dodecyl sulfate polyacrylamide gel electrophoresis, and the gel is made of polymerization of acrylamide and cross-linking of NN-methyl bis acrylamide. And the percentage of a gel can be modulated by modulating the acrylamide and the bis acrylamide ratio. And it all depends because this gel has the molecular sieves from which the protein moves. So depending upon the size of a protein, we can decide a gel. So for example, we have for example, 25 to 100 kilodalton protein, so 8% gel is used. And subsequently, the gel size is adjusted according to the protein size, because because this ratio also affects the sieves of a gel from which the protein travel. So, and the presence of SDS that I mentioned earlier, which is a potent anionic detergent, makes sure that all denatured protein will be coated with an equal charge to mass ratio. And larger protein travels slower in comparison to smaller protein due to retarding property of porous gel, and which is the basis of this polyacrylamide gel, where the separation is mainly because of the differences in the molecular weight. So if you see here, in the electrophoresis, if these are the smaller, they travel more, and the larger protein travel less, and they are found in the upper portion of the gel. So, and another very important property is of a gel of this gel is two different size of a gel. We have a stacking gel and a resolving gel. So, and stacking gel is a basic in nature, while the resolving gel, which is lower, stacking gel is acidic, and the resolving gel is basic. So a stacking gel stacks protein into a narrow band to allow protein to enter the resolving gel at the same time, which is made possible due to bigger pore size and acidity. So basically, the idea is that whenever we load a protein here, and once we load a protein here, the proteins are then separated based on the molecular weight, and we want to make sure that the resolution of this protein occurs at the same time. So we want to stack them together. Here is a boundary between the stacking gel and the resolving gel. So we want to stack them together so that when the separations start, they separate at the same time, and so we get a proper resolution. So this difference in gel is there. So, and that is once it is in resolving, which has a smaller pore size and basicity, the resolving gel, where the separation of the protein occurs. So this is where the stacking occur, and this is where the resolving occur. And generally is a classical electrophoresis from cathode to anode, and we also use a running buffer, which consists of a trace, glycine, and SDS at pH 8.3. So once coming back to whatever two parameters, just like loading, equally loading of the protein, and then separation according to the molecular weight. And third, foremost important part of the Western blot is transfer. So just consider this is a gel where all the proteins are loaded, and they are separated on the basis of molecular weight. Now they need to be transferred on a gel. And this Western blot electrophoresis transfer of a gel occur in a suitable membrane. And this membrane could be nitrocellulose membrane or PVDF membrane. And this membrane have a high protein binding affinity, and it immobilizes protein, and it is compatible with the detection method. So just for example, the arrangement that we make here is similar to similar of electrophoresis setup only, but here is that we keep a gel in the middle and then membrane, and then we use filter patterns sponge to make sure, and there is an anode and a cathode. So this is a classical electrophoresis setup where the protein from a gel is transferred to the membrane. And this is a basic definition of a blotting. Blotting means when you transfer any molecular content to the membrane. And in case of a Western blot, it is a protein. And this transfer buffer is constituted of 25 millimolar trace, glycine, and methanol at pH 8.3. And methanol is important for blotting, as it increases the hydrophobicity of a protein and facilitates the release of SDS, both of which increases the absorption of a protein onto the membrane. So the last step is antibody probing. So once the protein is loaded, and once it's loaded, then it is run on a gel, and the protein are separated according to molecular weight. And once the protein are separated according to the molecular weight, it is transferred into the membrane by the process of a blotting. And when we got a membrane, when we have a protein, so then we probe it, and we probe it by using a different methodology, where we first block the membrane. Blocking of a membrane is actually meant to block all the non-specific site. For example, this is our target of interest. For example, we are probing a CBS, a protein, and we want to mask the other side. So blocking buffer protein include either casein from a powdered milk or bovine serum albumin. So these actually mask the entire membrane, but the specificity of a primary antibody actually outcompetes the blocking, and it binds with the target on. And the purpose of blocking is that this membrane is highly sensitive of protein binding. So we don't want any non-specific or a false positive result. So we block the membrane first, and and as I mentioned you, the different protein like casein from a powdered milk or bovine serum albumin are generally used for this purpose. After this, the primary antibody binding detects the presence of a target protein on the membrane. For example, if we are probing, say, just as I'm in CBS, then we take an anti-CBS and primary antibody binds. And once the primary antibody binds, then we also follow a washing procedure, not mentioned here. We thoroughly wash with either a PBST or PBST washing buffer. And then there is a secondary antibody is incubated. Generally, what we do in our lab, that depending upon the antibody specificity, the incubation period can be 2 hours at room temperature, or 4 degrees at overnight at 4 degrees Celsius. And the secondary, generally, this secondary antibody is incubated for 2 hours. And it binds the target protein by binding the constant region of a primary antibody. And then we detect, because here the secondary are conjugated with the enzyme. Either it is a horseradish peroxidase or alkaline phosphatase. And then we use a substrate. For example, there is a substrate called luminol or DAP, and this give a color product. So this method is called chemiluminescence method. This method uses substrate that react with an enzyme conjugated secondary antibody, and these enzymes are either, as I mentioned, the horseradish peroxidase or alkaline phosphatase, and the enzyme mediated reaction produces a light that is recorded by imaging system. So this is an example of one of our experiment that we learned. So we have done in our lab. So generally we use a molecular ladder or molecular marker where we have a known molecular weight. And this known molecular weight that, for example, we then calibrate it with our running. So here it is this is a not transparent as well. This is just a gel where the proteins are run, and it is stained with comessi, and comessi binds with this protein. And here we can see the separation of a different molecular weight. It is not a Western blot yet. It is just a gel staining, but we can clearly see there is a almost equal loading in this. And then we probe it. Then we transfer this this gel in a blot, and then we probe it with our antibody of interest, for example, CBS. You can clearly see that CBS is this is the experiment where we given animal hypoxia for 7 days, 1 day, and 3 days. And this is a normal condition where there is no hypoxia. And we have, this is a brain tissue homogeneity. And after cell lysis and sample equilibration, this Western blot is developed. And these are the standard molecular marker that we know from this marker, protein marker set, that these are the molecular weight. And we know our antibody of interest molecular weight, and then we blot it. And we also calibrate it with a housekeeping gene. For example, beta actin is a housekeeping gene, which generally, their expression is not changed. So once we have a protein of interest blot, and once we have a housekeeping protein blot, then we normalize it to get the expression. So here we clearly see that the expression is changing in a day-dependent manner. So this is how Western blot give a lot of information about protein expression, but generally the standardization and titration of experiment is very important, because there has a lot of troubleshooting, because it is not as simple on bench as it sounds. There are many trouble and problem that we get, and you can troubleshoot it on depending upon the blots that you get. There are many times you have a smiling of a bands. There are many times you have multiple bands. There are many times where you have a high background. There are many times where an absence of a band. So we have to understand each and what are the conditions in which these things are happening. For example, smiling of band is most probably the main reason is voltage is too high, because generally when the electrophoresis is done, a constant voltage is provided to run the gel. If voltage is too high, then it is directly proportional to resistance and a temperature. So the temperature of the buffer may increase. The gel mobility is hampered. Or some other reason is that the volume of loading sample is too large. If the volume of loading sample is too large, it can also result in smiling of a band. And this is how you can titrate. For example, if you get this kind of a band, and to get a perfect this kind of a band, you have to understand, we have to see whether the voltage is given properly, or whether there is a sample is loaded properly. And if you get multiple band like this, and it is always suggested to get a single band when you are doing a Western analysis, to avoid false positive or false negative data. See, the presence of a multiple band suggests that the non-specific binding of antibodies, or it could be because a polyclonal antibody may be used, or it may be because of high concentration of a protein. So we have to see whether for example, generally the protein ratio is a 1:1000, or 1:10,000 primary antibody, or generally is for actin is more 1:50,000. So we have to see and calibrate and titrate our experiment according to our protein of interest and antibody. And if we see this kind of gel, then we can go into next experiment and we adjust few things. And if you get a high background like this, where nothing is clear and there is a dark, it's because of blocking. As I explained earlier, that blocking is the masking of antigen using different like casein or BSA. And yeah, it could also be because of excess antibody concentration, or because of a dry membrane. The membrane that I mentioned earlier, like nitrocellulose PVDF, has to be moistened all the time. If it is a dry, then the protein blotting is not proper. And these are the reason of high background. And sometimes you don't see anything, like the absence of a band. Even you loaded a protein properly and there's the absence of band. This most probably is because of poor transfer efficiency. So you may have a protein on a gel, but it is not transferred on the membrane. So we have some step like Ponceau staining. We stain with Ponceau and see whether the gel is properly transferred or not. So there are many other cases. So you have to titrate your Western blot. And the application of Western blot is many, as I mentioned, that is extensively used in biochemistry and molecular biology research to quantitate protein, to identify protein, to find a post-translational modification. And in Western blotting is also routinely used the verification of protein production after cloning. It is used in medical diagnostic. Few other things that is not written here is also, for example, these days in doping case also Western blots are used. So this is entire Western blotting that I've explained. If you have any question, then kindly. Hello. Hello.
Thank you very
Yes.
much. Thank you very much. Thank you for this excellent presentation. Although you have cleared all the doubts related to the Western blot method, and this is a very good technique for the determination and identification, even the quantification of the proteins,
Yes.
upregulation and the downregulation after treatment.
Yes.
But out of the queries, I'm having one question, and I think this is a genuine question related to the Western blot method, because you would be agree with the condition that the antibodies which are associated in the Western blotting method, it has highly cost. So can you give some way, means how we can procure these antibodies means by giving the so much of cost?
Okay. So if I understand correctly, you want to understand, you ask how we choose antibody, right?
No. Actually, my question is, suppose if I'm going to purchase the antibodies,
okay,
the cost of the antibodies starting from the 30,000 or 35, or in some time it's reaching to the 60,000 also. So can you tell me the way, means how can procure these antibodies without putting the high pressure on my pocket?
Okay. Because I am lucky to use AbChem antibody in my lab, and generally the technology and a quality control is very important. Is very important in Western blotting. You don't want because you don't want a false positive or false negative result. So I think though people isolate this antibody, they do in the lab, they produce maybe 20 years before, but I haven't done myself antibody isolation. But I suggest that first of all, the cost is an important factor. I agree, but it also brings trust of the reviewer and avoidance of repetitive experiment. Because if you use a CST or cell CST or AbChem antibody, then definitely you are going to get a blot in 2 or 3 times, or maybe first time if your hand is good, first 2 or 3 times. So the cost looks high at the first hand, but if you put the labor and the time. So I think even the reviewers these days, they just make sure that you get it from the good companies to avoid. So there are ways to do it, maybe at pilot experiment. But I strongly believe that a good quality of antibody is very important for a good assay.
Right, sir. Thank you very much.
I request all the participants, if they are having any questions, kindly type them in the chat box or raise your hand. Gaurav sir, hello.
Hello, yes, sir.
Sir, there is one question from Ms. Vandana Thakur. Staining of bands and quantification of band. And first question is this: staining of bands and quantification of band.
Okay, quantification of a band is generally that we do is using a ImageJ software. We actually what we do is that we select a area where the blot is, and we subtract it with the background, and we repeat this with the housekeeping protein that we do, and then we normalize it with protein of interest. If you remember the slide I have shown you, there are CBS and actin. So first of all, I quantitate actin with the help of the ImageJ software, which measures the pixel size, dependent intensities, and then we normalize it with the with our experimental setup. For example, under a hypothesis that that the housekeeping is not changed in the experiment, and the protein of interest is changed. Yes. And second question
The second question is: can we use BCA public biuret protein assay instead of Bradford?
Yeah, we can use, definitely we can use. Definitely you can use. It has a,
okay, sir. Okay, continue, sir.
Because it has a different dye, which gives after combining with protein a different adduct, and which can be a spectrophotoly measured. So we can definitely use it, and the BCA cost is perhaps more than the Bradford, I think.
Next question is, sir: can coronavirus be detected by Western blot?
Yes, yeah, it can be definitely detected by Western blot, because if you have for example, if you know a COVID spike protein, and if you have antibody of a COVID spike protein, and which is a very unique, and then you can detect. So I haven't gone through the papers of this, but definitely by the if you have a specific spike protein you can definitely detect.
Okay, sir. And next question is there any process to identify the active site of protein experimentally?
Active site of a protein experimentally, I think Western blot, with Western blot, we cannot determine active site of protein. There are other kind of experiment like site-directed mutagenesis or other experiment, which is beyond the scope of this talk. So active site is Western blot, and active site is, I think, is not related. There are other technique and procedure and other structural features that you can do.
Yes, sir. Participants, any other question? Is there any other question? Sir, one more question is there
Yes.
from Dr. Janki Patel. Any other way of probing other than using antibodies?
Without antibodies, you want to probe a protein?
Yes, sir.
Generally, people use, if you want to understand, for example, you can use this RT-PCR or gene-based technologies also. For example, PCR and other things where you can know the expression of a gene. But for protein quantity, these are the gold standard experiment.
Okay, sir. Thank you. I think there are no more questions. Rupali madam, kindly conclude. Rupali madam.
Yes, sir. Thank you, sir, for such a wonderful session. And we are very grateful for you, for your effort and time you took to share your knowledge and experiences with us. Thank you, sir. Thank you for your contribution. Nitin, sir, would you like to say something?
Nitin sir.
Yes, Rupali madam, I would like. Thank you. Thank you, Gaurav sir, and thank you, Rupali madam, for this opportunity. Actually, this was the excellent session of the FTP. Along with this, I am having something to share with the faculty member who are joining this faculty development program. As you know, the all faculty member of the different institutes, the Department of Pharmaceutical Technology of MIT Meerut is continuously working to enhance and strengthen the quality of the research and development in the departments. So in the last few years, we have developed few of our laboratory to strengthen the quality of the research and development, and we believe that the resources which we have developed in our laboratory, that should be available to the faculty member of the other institute as well. So with the same objective, I would like to take this opportunity to offer the benefit of our resources and the specialized laboratory to the faculty member of the other institute as well as. So Rupali madam, can you share the flyer?
Yes, sir, sure, I'm sharing.
Right, thank you, madam. So with the same objective, we have developed our two laboratory specifically. First laboratory is the Center for the Preclinical Research. Currently, we have received the CPC approval for conducting the research and development for the commercial project as well. And we are already handling, my faculty member are already handling the industrial project for total overall cost of the 20 lakh. So I believe that my laboratory of this preclinical research should be available to the faculty member of the other institute as well as, and the researcher who are working on for the doctorate program or who are going for the post-doctorate program. It doesn't mean that you're incapable of doing your research in your laboratory, but sometimes some constraint like if there is a time constraint or if there is an infrastructure constraint, so you can connect with us and we will provide you our laboratory for conduction of your research. So this is one of the laboratories, the Center for the Preclinical Research. We are having this infrastructure. We are having the many kind of the laboratory animal like guinea pig, mice, rat, and the rabbits. We are having the approval of those animals and can allow you or the faculty member with the consent of the CPC approval to conduct your research in our laboratory. So in the laboratory, we are conducting the toxicity study, the acute, chronic, and subchronic, all the oral and the topical or any kind of the toxicity study you can perform. The efficacy study you can perform, the pharmacokinetic study, the BA/BE study, the IV-IC correlations, or the any kind of the model development. If you need the support of any kind of model, like currently we have developed the Alzheimer model, the diabetes model, the remyelinations for the Alzheimer disease, that or the COPD model we also develop in our laboratory, and the process is continuously going on, and even we are on the learning phase as well. But my laboratory can provide you some specialized type of the facilities, and I believe the research out of this laboratory will enhance the quality of your research publication and the IPR protection. Rupali madam, can we have the look of the second flyer? Right, thank you. Similarly, Now we are launching another laboratory in the field of the pharmaceutical chemistry, and the name of the laboratory is the Center for the Chemical Research and the Development. Similarly, here you are going to be perform the any chemical type of, means if you want to synthesize any chemical moieties, or if you want to perform any QSAR studies of your prepared chemical moieties. So these kind of the research, we are having some specialized software related to the QSAR. We are having the sophisticated apparatus and instrument for conduction of your chemical preparation of the chemical research. So these kind of the research you can perform in the Center for the Chemical CCCD. So thank you, Rupali madam. And at the same time, I would like to request Garima madam, who is serving in the Department of Pharmacy as the manager of these kind of the work. Garima madam. Garima madam, are you there? Can you switch on? Rupali madam, can you allow Garima madam to say a few words?
Sir, I'm here, sir. Can you hear me?
Yes.
Good morning, everyone. Thank you, Nitin sir, for introducing both our Center of Excellence with the participants. I would say that both our Centers of Excellence, the Center for Preclinical Research as well as CCRD, are a feather in MIET's cap. And these centers both serve to boost teaching, training, and research facilities. So, and I am also very proud to say that we have executed over 20 plus projects in the past 6 to 8 months only, and we have about 20 more projects in pipeline. So yes, and our faculty, the average experience of our faculty is 15 plus years. So this makes us one of the best teams in the region. So we would be glad to offer, like Nitin sir said, offer the facilities to anyone who would like to make use of them, and we can help you out advance your research. That's all from my side. You can always contact me if you have any questions. We will be circulating the both the flyers on the WhatsApp group as well as the Telegram group. And post that, if you do have any questions, you can always contact me on WhatsApp or a call on the numbers given and the email ID given. Thank you, sir. Over to you.
Right. Thank you, Gauri madam. Again, requesting all the faculty members, means this will be the win-to-win situation for everyone. If you are having any ideas or if you are having any constraint in your going on research program, so definitely the Department of the Pharmaceutical Technology can give you the support in terms of the instrumentation, or in terms of the crystallization of your the IPR. So thank you very much, Rupali madam. Thank you. Please continue.
For the participants, this session has finished. For next session, let us meet at 11:50. Exactly at 11:50, let us meet. So that we should start the next session. Okay, Rupali madam.
Yes, sir. The link for the attendance and feedback will be shared at the WhatsApp group.
That will be shared shortly. And for this first session and second session, we will be starting at 11:50. Thank you.
Vipin sir, should we start?
Yes, madam.
Okay, so thank you. A very good morning to all once again. Today, we are starting with the session 2 of day 4 for Atal sponsored 5 days FDP program on application of biotechnological and analytical tools in pharmaceutical research. So let's start our technical session round with Dr. Gaurav. But first of all, I would like to take the opportunity to introduce Dr. Gaurav to you all. Sir has done his PhD in year 2016 in neurobiology and life sciences from Defense Institute of Physiology and Allied Sciences. He has been the National Postdoctoral Fellow, School of Life Sciences, Jawaharlal University, New Delhi. Currently, ICMR Research Associate, Department of Peptide and Proteomics, Defense Institute of Physiology and Allied Sciences. Sir has been advancedly trained in the CAGAL, Advanced Neuroscience Training in Brain Homeostasis and Neurovascular Coupling from Bordeaux, France in 2019. He has also been trained in Advanced Wellcome Trust course on System Biology and Functional Genomics from Sanger Institute, Cambridge, UK. Molecular neuroanatomy course he has done from Okinawa Institute of Sciences and Technology, Japan in year 2011. Sir has made several national and international publications in good impact factor journals. He is also the member of International Society of Cerebral Blood Flow and Metabolism and also the member of American Chemical Society. So today he will going to deal with the topic ELISA as a platform for immunogenicity assay in drug development. Welcome you, sir.
Thank you for a very kind introduction. And let me share the screen. So this is another immunoassay, and which is different from Western blot in terms that it is very sensitive, cost-wise, it's cheap, it's quick and convenient. And this also detects the native form of protein, because in Western blot, what we have seen that we break the disulfide bond and another thing. So this is very interesting and very clinically and biomedically relevant, which is used in research also and for the diagnostic purpose also. So let me begin with ELISA. So this, the full form of ELISA is enzyme-linked immunosorbent assay, and it detects and quantifies a soluble substance. So not only protein, it also quantify peptides, antibodies, and hormones. So if I so in ELISA, antigen is a very important step of ELISA. Is antigen is immobilized in a solid surface, a microplate, as this you can see, and is then complexed with antibody, and it's linked with a reporter enzyme, and which is then detected. And the most crucial element of ELISA is highly specific antigen and antibody interaction. And this is actually common to all the immunoassay where this phenomena is exploited to detect a antigen and not only detect but also quantify antigen. So ELISAs are generally performed on a polystyrene plates, typically 96 well plate. And these 96 well plate are generally, there are commercially available ELISA plates also, and there are protocol and procedure in which you can also design your own ELISA plate using NUNC plates and then following different steps. The most important step in ELISAs are coating, which are done either with antigen or antibody. And it is a direct or indirect mobilization of antigen to the surface of polystyrene microplate well. So here what happens is that we use a coating buffer, little alkaline, 7.4 to 9.5. and it affects the steric structure of a protein, antibody, and analyte binding, and thus cause immobilization of this antigen on the polystyrene surfaces. And following coating, we also block this, we follow the blocking step. And here the purpose is clear to avoid any non-specific binding of antibodies. And generally BSA, 1% BSAs are used. As I mentioned, the addition of irrelevant protein or other molecule to cover all unsaturated surface of binding site of the microplate to avoid any false positive or a false negative result. And following the blocking, we detect it, and detection is done with the help of enzyme, horse radius peroxidase or alkaline phosphatase, and substrate that we add. TMB binds with the HRP, and there is another NNP that binds with alkaline phosphatase. And the byproduct, the colored byproduct based on its observation is actually detected by a spectrophotometer, and this detection of spectrophotometer is finally quantified. So the idea is that you take a polystyrene plate and you coat it with your desired antigen or antibody, and then you block, and then you use detect and read. So this is somehow analogous to Western blot, but little different, that I will explain in my subsequent slide. So there are different type of ELISA based on the antibody and antigen interaction. There are 4 major type of ELISAs: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. So I will explain each in detail. So actually in direct ELISA, you see the antigen is coated on the plate, and the first step is, as I mentioned, the first step is coating of antigen. Involves adding antigen to the plate, which is incubated for an hour or incubated 4 degrees overnight. It depends upon the antigen, and it differs because there are many commercially available kit also. Or if you are designing your own ELISA, then you have to titrate according to your need. Next step is washing. Next step is blocking, followed by washing the plate, and using antigen like PSA, ovalamine protein, or other animal protein. Following a selected enzyme conjugated primary detect antibody, for example, alkaline phosphatase or horse HRPs, which are generally conjugated with the primary antibodies. And finally, if in ideal case, if this reaction goes well, when the antigen binds with the primary antibody, and the substrate is added and it changes color, and it's detected in ELISA plate. So this is the entire scheme of story for direct ELISA. And the advantage of direct ELISA, it eliminates secondary antibody cross-reactivity. It has the fewer steps, and it is a very rapid. And disadvantage, it is very low sensitive compared to other ELISA, and it's a high cost of reaction. So based on the experimental needs, based on the type of samples and analyzed, different type of ELISAs are generally used. So in second type of ELISA is actually indirect ELISA, and its first step is similar to the direct ELISA. And here what you see is that there are 2 types of antibodies that are used: the primary antibody and a secondary antibody. And here the conjugate are with the secondary antibody, the enzyme conjugate. So in a perfect case, when the indirect antibody, the antigen binds with primary antibody and secondary antibody, and so the substrate gives color. So here the main difference is addition of secondary antibody, and it has an advantage. It is higher sensitivity than compared to direct ELISA, and it is considered less expensive and more flexible to many possible primary antibodies that can be used. And there is a risk of cross-reactivity between the secondary detection antibody. Here also the steps are carefully managed. The washing step is very careful to avoid any kind of false detection. And if we go in the next step is sandwich ELISA. So here the addition of a capture antibody to the plate. Here actually the capture antibodies are on the plate, and these are incubated first. And once the coating step is complete, the plate are washed with PBS and then buffer blocked with BSA. After the buffer washes, the antigen of interest is then added to the plate to bind the capture antibody and incubated for around 90 minutes to at 37 degrees Celsius. A plate is then rewashed and the primary detection antibody is then added. Following a washing secondary antibody is added. Following the wash step, the addition of a substrate and detection of a color changes. So the idea is there's a method of washing each time is to avoid any non-specificity, and only a very strong binding with antibodies and antigen and primary and secondary boundary are only remains, and the rest of the samples are blocked, and the reading gives a clear idea about the detection. Sandwich ELISA has the highest sensitivity amongst all ELISA types. The major disadvantage of this ELISAs are time and expense, and necessary use of a match pair. For example, divalent or multivalent antigen, which we have to decide. So it is a little cumbersome to design this experiment. And another is a comparative ELISA. So it is a little different from other ELISA. Here, what happens is if you see here, the antigens are pre-coated, and that is and antibodies are also provided with a conjugate, and the samples are added. So the sample antibodies and the conjugate antibodies compete with each other. So in an ideal case, when the sample has an antibody, then the detection color generally decreases, because it competes with the already provided primary antibody with the conjugate. And in case where it is absent, the only provided antibody binds with the antigen and it gives the color. So if I explain you more details, is that this type of ELISA utilizes two specific antibodies, an enzyme conjugate antibody and another antibody present in a test serum. Combining two antibodies into the wells will allow for a competition for binding to the antigen. So the positive says is absence of a color or decrease in a color indicate a positive test and the presence of antibody in a test serum. A negative test is the presence of a color change, means the test is negative because the enzyme conjugate antibody bound to the antigen, not to the antibody of a test serum. So advantage is a less sample purification needed. It can measure a large range of antigen in a given sample, and its disadvantage is the low specificity and cannot be used in a dilute sample. So based on the approaches and antibody-antigen interaction, these four types of ELISAs are here that I explained. So how do we quantitate? ELISA estimation is done generally with a standard where we know the antibody concentration and the sample. These are generally provided in a kit or we know it. So we make a serial dilution and then a standard curve, and from the standard curve that we make, we generally detect from our samples. First, the standard curve is made, and you see the curve. As I explained, that in competitive ELISA is a little different, because if there is a less concentration of a sample, there will be more detection of a color, and contrary to the non-competitive ELISA. And then the sample reading is done, and with the proper dilution, it is a quantitative. So there are many factors which interferes with ELISA. For example, that we have to take into account while doing ELISA. It's like plate assay. the shape and quantity of a well, the material of a plate, a potential reactivation or even or uneven coating is very important in getting the result. We have to check the buffer, pH and contamination as all biological reaction takes place in solutions. So solutions, pH and its composition is very important, not only in ELISA, but in any molecular biology experiment. And capture and detection antibody, the incubation time, temperature should be titrated according to the experiment. And the blocking buffer, cross-reactivity, target antigen, enzyme conjugate, washes, it has to be proper washes, the frequency, the volume, duration, and composition of washes. Substrate is another important factor, and detection and the reader, and human error, of course. So these are the various variables that you have to take in account while doing an ELISA, or to make sure each variable has to be controlled properly, so that we can quantify in a very accurately with no any false positive results. So ELISAs are these days are used in many kit in detection of IgM and IgG antibody post-COVID infection, and they are very, as I explained, they are quick and convenient, and direct or indirect ELISA are used to detect these IgM and IgG antibodies, and which can give a clear idea about the antibody produced in infected or a non-infected or in asymptomatic patients. So there are many applications of ELISA in diagnostic and many diagnostic tests. It measures the presence of antibodies in the blood. It detects and estimate level of tumor marker as well, hormone level as well, and tracking disease outbreak. Just I mentioned that in a recent pandemic, the ELISA kits are very relevant in determining the IgM and IgG concentration in a patient, and detecting past exposure also. It also donate blood for a possible virus contaminate. It detects drug abuse. So there are many advantages of this quick and rapid. So though it is highly sensitive, but we have to control this. And compared to Western blot, it is a little, it's a more high throughput. So this is mainly my experiment, my explanation to ELISA. I hand over for more questions. Hello.
If participants have any question, they can raise their hand or post their queries in chat box. So there is one question: was buffer of one ELISA kit, can it be used in another test? For example, wash buffer of anti-MPO, can it be used in IL-6?
Okay, you're saying the wash buffer of one ELISA kit can be used for another ELISA kit?
Yes.
So
yes.
generally, a wash buffer are having a same composition, but the idea is you have to titrate it according to the antibody needs, according to the experiment need. So maybe the wash buffer can be used, but the number of washes, the time of washes has to be titrated for each antibody or each different experiment.
Participants can post their query in chat box or they can raise their hands. So there's a question that what factors should you consider when trying to choose an ELISA test?
What factors we choose when we decide to use ELISA? Okay, so generally ELISAs are done in a diluted samples, diluted samples, and the idea is to have a more when we want a fast, sensitive, reliable detection of antigen. So we choose ELISA over any other antibody detection test. So the idea is to, in any case, ELISA is done to know about the antigen, and the basic thing is that if you compare ELISA with other technique like protein, Western blot, the Western blot is considered a final stumble. For example, there may be a case when ELISA can give a negative result, so you finally confirm with the Western blot. But to get an idea, for example, plasma samples, for example, diagnostic purposes, for example, large scale detection. So we choose this ELISA kit. It's very handy. It requires less expensive instrument. So these are the main factors.
Okay, so one question is how to express the final results of the ELISA.
How to express the final result of ELISA is a protein concentration per ml, right?
Yes.
Is protein concentration per ml that we have to detect. Actually, it requires making of standard curve and detection. So exactly we have to see by the kit, but it's mainly protein concentration per ml. Per picogram is more relevant because it's like a concentration per picogram and concentration of a protein per volume. So this volume could be picogram or more or something.
So next question is that which kind of enzymes is generally used in ELISA?
So there are two kind of enzyme that I mentioned: horse radish HRP and alkaline phosphatase. This HRP binds with a substrate called TMB and give at 605 absorption, and alkaline phosphate bind with P and NPP. So these are the mainly two things: HRP and AP.
So another question is that why sulfuric acid is generally used to stop solution? Yes, yes, sir.
So it changes the pH. Sulfuric acid is used because it changes the pH. So maybe.
So next is that is it possible to perform the ELISA if standard or positive control is not available?
It is not recommended. It can give. It is not totally recommended because when you are doing your experiment, you have to make your standard each time. Because there are many conditions, many variables. I mentioned many variables. So your standard and your experiment should be controlled. The variability should not differ. Because if you have done some, maybe the water quality is different, the molecule quality is different, temperature is different. Even the humidity affects the experiment, so it is not totally not.
Okay. So one question is there any effect to the antibody titer in hemolyzed serum samples?
Antibody?
Antibody titer, I think it's related to titration. Is there any effect to the antibody titer in hemolyzed serum samples?
Hemolyzed antibody titer hemolyzed serum sample.
Yes, sir.
Ma'am, I can't comment on this because I actually haven't done this, so it's totally.
Okay. Okay. No issues, sir. No issues. Okay. So that's all for now. There are no more questions. So, Vipin sir, can we conclude it?
Yes, madam, we can conclude.
Okay. So thanks a lot, sir, for taking your time to speak to our audiences on this very innovative kind of topic that is ELISA. And I felt this whole thing will help us a lot in future. Thank you, sir. Thanks a lot.
Thank you, Dr. Gaurav. Participants, we will, for the last session, we will meet at 2:50. Kindly join at 2:50 p.m. And last session, today's last session of Dr. Anand Narayan Bhatt, we will be meeting at 2:50. Then tomorrow there will be hands-on session on ELISA as well as maybe ELISA or Western blot and flow cytometer. So for now, we have to meet at 2:50 p.m. Okay. Rupali madam?
Yes, sir. The attendance and feedback link for the participants will be shared at the WhatsApp group. Please do fill it kindly. Thank you all. Thank you for now.
Thank you. Thank you.
Vipin sir. Vipin sir, am I audible?
Yes, sir.
Rupali ma'am, Anand sir will be joining in next 5 minutes.
Okay, sir.
Okay. Anand sir, good afternoon.
Good afternoon, Nitin. Can you see me?
Yes, sir. You're good.
Okay.
Should we start, sir?
Yes, I think I'm ready. We can start.
Okay, sir. Thank you. A very good afternoon to one and all present over here, and welcome back again for the last session of today. And I'm very overwhelmed for taking this opportunity to welcome Dr. Anant Narayan Bhatt, Scientist F in Mass Delhi. Sir has completed his MSc Biochemistry in June 2000 from Dr. RML Awadh University, Faizabad, India. PhD Biochemistry in June 2006 from Molecular and Structural Biology Division, Central Drug Research Institute, Lucknow, India, and Dr. RML Awadh University, Faizabad, India. Sir has joined Institute of Nuclear Medicine and Allied Sciences, Delhi, in year 2005 as Scientist B and currently serving as Scientist F for the same organization. Sir has bagged several awards and honors, and some glimpse of that are European Molecular Biology Organization Fellowship Award from European Organization in 2012, Fulbright Fellowship Award from US India Educational Foundation in 2013, Laboratory Group Technology Award in 2019, Technology Day Oration Award in 2019, and Science Day Oration Award in 2020. He has published several research papers, abstracts, and book chapters in high-impact journals. Another feather in his cap is the development of the anti-COVID drug 2-deoxy-D-glucose with his team in DRDO. It's a privilege, sir, to have you here among us all for giving a talk on the topic flow cytometry, breaking bottleneck in the drug discovery and development. Over to you, sir.
Thank you. Thank you very much, Dr. Rupali, for a generous introduction. So today we are going to talk about the flow cytometry and its principle and how we can use it in our daily research work. So my screen is visible?
No, sir, not right now.
Sharing is paused. It says resume share. It shows some problem. I mean, it says your screen sharing is paused.
Okay. Sir, please do not move to some other screen while sharing.
Yeah,
if
I'm
it is
not.
so, then only it is say that.
Yeah, I'm not moving to any other screen. I'm just.
I think it will take a few moments, like 30 or 40 seconds, because it may be due to the lack of network from your side, sir.
Okay.
Just wait, sir.
Said I stop the video, then probably it may.
And sir, I think this is taking the unusual time. Just stop sharing your screen.
I just stopped. Who can share only host? Shall I click all panelists? Because I joined as a panelist. Advanced sharing option. Who can start sharing when someone else is sharing? Yeah, so I just clicked that.
It's
Is it visible
now. Yes,
Okay.
sir.
Okay. I think I joined as a panelist, so probably it was only visible, allowed for host. So we are going to talk today about the flow cytometry principle and its application, how we can use this technique in our day-to-day research and drug development program predominantly. So before going to the next topic, I mean, what basically we should understand that what is basically flow and flow cytometry, what does it mean? So here, I mean, unlike other techniques where see what basically when we are talking about cytometry, basically we are emphasizing that we are measuring whatever changes are happening in cells. That is why it says cytometry. So and we are measuring the cells or the changes in these cells when they are in the flow. So that is why this technique is called flow cytometry. So we can measure n number of parameters and a characteristic of cells when they are in the flow. So let's say in microscopy, when we measure the cells when they are static, or majority of the techniques when we measure the cells, they are in a static condition. Here we can measure the cells when they are in I mean flowing condition. So for blood cells it becomes very easy. I mean that for the blood cells which are actually in flowing conditions, when we measure the cells in flow cytometry, the blood cells get the actual atmosphere for its measurement. And you know, like flow cytometry started initially for measuring the blood parameters only. Later people realized that the culture cells also we can bring in the suspension form, and by bringing them in suspension form you can analyze a lot of things by staining that with fluorescence fluorescence dyes. So measuring both intrinsic and extrinsic properties of cells while cell is in a moving stream. So that is what called basically the flow cytometry. So how it is different than microscopy? You know, like in microscopy also we can analyze a lot of extrinsic properties, morphology of the cells, whether the cell is dying or it is still alive. So many things we can observe in flow microscopy. And by adding these fluorescence dyes also, which can bind to specific cellular organelles, or they can bind, like you can measure the ROS inside the cell. That also you can measure inside the cell using microscopy. But what is the limitation in microscopy, and that is why we need flow cytometry technique, is like it is a qualitative. It will not give you exact quantitative idea. But you know, like in microscopy, lot of new techniques has developed with the development of algorithm, where you can process your images and you can extract that information also. So you can convert the qualitative image into quantitative information, but that information will still be a localized information. You know, like suppose that when you set the cells in some petri dishes, what will happen basically, you know that locally what is happening in that area. Many a times when you'll observe the cells in petri dishes or flasks under the microscope, you will see that those some patches give a different information than rest of the microscope. But here what you do, you take out all the cells from those flasks or petri dishes, and then you bring them in suspension form. And when you run it on the flow cytometer, it gives the comprehensive holistic information of the sample, not just different information in different patches. So that way flow cytometry is better. It gives directly quantitative information. Then it is very good. Microscopy is very good for screening to find out times, etc. But when you know the exact time, this properties, these things these whatever change in the parameter or whatever new phenomena which is happening in the cell at this particular time, then you should go ahead and do the flow cytometry. That will give you the exact information of that particular thing, effect of drug, or the change in the cells for the betterment, or if you are designing, let's say, any anti-cancer drug, so it should cause more cell death. So you should know, after adding to the cells, the cell death is happening at what time point. So you can add the drug into the cells, and then you can monitor at different time point what is basically happening to the cells. So on the right side, you can see here that this is let's say it is a histogram on the in this panel. You can see that this is called histogram, where the signal will come in the flow cytometry, and majority of the signal is coming in this area. So most of the cells are focused in this area, but you can see like this is a small population having a different behavior than this large population of cells is having a different behavior. And this is called scatter or dot plot of the same thing, which you can see like these things are coming in different clusters of the cells. So like in whatever cell you have treated with the drug, or if you are analyzing the property, all the cell population exist in 3 different populations. So these information also can be easily, you can see in any given sample size, or in any given sample which is treated. So this is a very this gives very useful information from that perspective. It's not moving. Okay. Hello?
Yes, sir.
Yeah, actually it is not moving. I don't know.
Yes, sir. Your slides are not changing. Yes, now it's changing, sir.
Yeah, so these are different flow cytometers here. You can see here, this is a very old flow cytometer. It's called Fax Caliber. And then the lower one you can see here, this is actually it's called LSR2. And then this is a very small one which can be easily kept in the lab on the cells. This is a small size instrument that's called Accuray. And this is again another one which Fortesa, and then so many new instruments have come. So majority of these instruments can be actually can be used for in different labs for our purpose. Yeah. So basically, what happens, as I said, when you take out, like when you treat the cells, or when you are analyzing the cell should be supposed to be in the form of, in the on the petri dishes, or they should be seeded in the flask. But when you treat these cells with drugs, and when you want to analyze the effect of the drug on these cells, they should be scrubbed, or they should be trypsinized to bring them in the solution form. And then in PBS or saline or whatever buffer is recommended for your experiment, you resuspend the cells in those suspension form, and then you load on these, that comes in a flow tube, and then there is a nozzle where you fit this flow tube, and then it starts sucking the sample, and then that goes through. On the right side, you can see here, here on the right side, you can see here that this is like coming, this is coming from the sample, and this goes into this seed fluid in a nozzle stream, and here a single cell, it forms a stream of single cells. So this stream when goes, then laser comes from this side. laser will come from this side, and this laser that is specific, because if you have scanned your cells with some specific marker, then that will get excited on some specific wavelength which comes from the laser. So in the setting, you select that which wavelength you want to use for analysis of that specific parameter. So let's say if you want to use ROS, if you want to measure that whether your drug changes ROS or not, then you will select the blue laser. So it will fall on this, and then that will give the green color emission. So that will fall in the FDP window. So then you will be able to see that how much when the cells are passing through it, when laser falls, some cells may have very high amount of ROS, some cells may have very low amount of ROS, and that will show a different scatter plot, and that will show a different histogram. So you'll be able to measure that what is the level of ROS in that specific sample. There is some problem, I don't know, due to yeah, so yeah, so you can see here that when sample flow, very important thing, one more important thing, when you are analyzing the sample, then the speed of the sample should also be optimized. So you can see on the left side, the sample speed is slow, so single cells are going through it. When the sample size, the speed of the flow will be very high, what will happen? See, some of the thing, I mean, those samples which are measured when you are measuring just single change of the fluorescence, like ROS and other things, then high sample size is also fine. But when specifically when you are measuring the cell cycle pattern, then like very optimal sample flow is required. You can see the difference on the in the upper right side histogram and lower right side histogram. You can see the upper one is at a low sample, fine, very thin. So that shows that majority of the cells, when they were measured, then they were in the this very specific region, very specific region, and very accurate measurement of the sample you have received. But when you can see the sample on the right, the lower one, you can see that when the sample flow was very high, what happened, that the same this thing has gone a very wider CV. This is called CV. Variation is very high, and because when laser is passing through, many cells are analyzed at one go. When the sample stream will be high and the sample flow will be low, then what will happen? Every cell will get analyzed by the laser individually. So that is why your data will be more accurate, which you can see on the top side, on the top blue graph. But in the lower blue graph, you will see that the values, it is the same sample, but you can see the values change is happening at very fast pace. In the bracket, you can see that the values are given. So from 68.7%, the G1 population became 74.85%. So almost 6-7% change is happening. So that is why this CV becomes 7.79. So if CV is less than 3 or 4, then the data acquisition is very good. So this is very important, that we should acquire the data at appropriate sample speed. So here, actually, this is this gives. So there are when you will be measuring this, you will be seeing lot of many changes into this, many parameters. So I just want to highlight little bit what are these parameters. So the two major parameters in dot plot, you will see that they are called forward scatter cytogram and then side scatter cytogram. So this forward from where this forward scatter come and from where this side scatter comes. So forward scatter basically detects about the cell size. If the cell is bigger, then forward scatter will be higher. And if the cell is smaller, then the forward scatter will be smaller. The side scatter comes from the granularity of the cell. So if the cell will be having very high granularity, the side scatter will be high. And if the cell will be having low granularity, then the side scatter will be low. So you can, how you can differentiate between this? If you remember the basic experiment of Rutherford, which he did to show that the nucleus has some strong mass. So he bombarded the atom with alpha particle, and then alpha particle showed they did not go directly. They were there was a lot of side scattering of this alpha particles. So he said that it is because the atom's nucleus has a big mass, and that is why side scattering is happening. It is reflecting the path of the alpha particle bombardment. So you can get that from side scatter basically comes from the granularity. Because the cells are more granular, then you will have high side scattering, and if the cells are less granular, you will have less side scattering, and the forward scatter will come from the size of the cells. So based on that, actually, as I said, the flow cytometer was initially used for analyzing the blood parameters. So this is a WBC parameter. You know, when you process and analyze WBC, you get very mild, a small, what you call, the contamination of RBCs is already there. So you can see here, and RBC is very small as compared to rest of the WBC cells. So you can see on the left side, this is RBCs and debris and dead cells are there, which are very small. They have very small forward scatter as well as side scatter. But when the lymphocytes are little bigger, among the WBCs, lymphocytes are the smallest. So you can see here, the side scatter is also very small of this one, and forward scatter is also not that big. So this comes among the WBCs, this comes lowest here. Then if you see the mononucleomorphs, you can see here that they have little bigger size forward scatter, but their side scatter is not that high. So that is why they are showing low. But then if you go to the granulocytes, all these granulocytes which are there in the cells, in the WBCs, you can see they have high forward scatter as well as side scatter. So you can see, if you run the WBCs isolated from the blood, you can see the different cluster of the cells. This is a what you call dot plot of the WBC, and not measuring any fluorescence, not measuring any staining of it. It is just the simple forward and side scatter based on the extrinsic property, you can say, of the blood cells. So this is how it happens. Like inside the instrument, it looks like this. So you have different lasers, and different lasers will have different, for different kind of dye. As I said, for measuring this ROS, which is DCFDA or FITC, or FITC is like a very popular green dye. That is why the channel name itself is given FITC channel. Then there is a PE channel. So PE channel is basically which can trap the red colors. red color fluorescence. So that is called PE channel. So your PI, propydium iodide measurement, which is used for cell cycle, or PE dye itself, if you have stained anything specific protein and did immunophenotyping, so you immunofluorescence if you have done, so what you I mean, you can tag with a specific antibody and then secondary antibody which has PE or any red marker or any green marker, so like FITC and all, and then you put the secondary antibody and stain that and then run the cells into this. So it will tell you the level of that specific protein in that specific cell. So that can be done by selecting different kind of lasers in your setting. So you should know like what specific protein when you are measuring, and what your antibody is, what fluorochrome your antibody has. So that will come in, for this setting of the experiment. Those things will be essentially required. That is one thing. And then second thing, like when you are using direct fluorophore, let's say as I am telling that propydium iodide can go and bind to DNA. So you can do the cell cycle measurement by using propydium iodide. So you need to select P channel for that. So based on different content of, like in G1, the DNA content is low. In the S phase, the DNA content will increase from G1 to slowly, I mean, different level of, in S phase, you will get different level of DNA content in different cells because they are in different phases of S. So they may synthesize like 1.1, let's say from 1.1 to 2, because in G2 or M phase, the cell will have double amount of DNA. So, so anywhere between 1.1 to 1.9 will come in the S phase. And when the DNA content will become 2, then it will come in G2 or M phase. So that's how, we will discuss in next slides in application that how to do cell cycle. So that is how it gives you the complete chromatogram or histogram of the cell cycle analysis in that. So based on different dye content, different DNA content, the PI binding will be more, and then that will be reflected in the PE or PI channel, red channel. So when you have stained the cells with different kind of dye, you can see here the FITC shows in green color with blue laser, and PE is a different, which is anywhere between red and yellow. So P shows here. Then there are a lot of red color dye comes, and then P is very dark red, and then there are new dye has come, Psi5 and then Persi-P. They are like anywhere between yellow and red. So they show the color in the orange region. Somebody want to ask some question? Hello.
Answer, we will have the question answer at the end of the presentation.
Okay. So so this when you are measuring many colors, many things at one time only, then you can see here FITC. If you have, let's say, if you have stained some cells with PE, the same cells with PE and FITC both, PE for some other parameter and FITC for some other parameter. So you are analyzing two parameters in the same cells. So what will happen? Some signal will, go in the range of PE, and some P signal will go in the range of FITC. So this is called a spillover of the signal. So for that, those things, a compensation is required. So you need to stain cells with only FITC, and you need to stain some cells with only PE, and you run this separately also. And when you run this separately, that will be a reference sample for your experiments. And then using these separate staining, when you in your test sample, when both the dyes were kept together, when you will process those data, then this will use this use reference samples, and it will take care of the spillovers, signals, and that process is called compensation. So when you are measuring many dyes at a time, this compensation is very important thing, which takes care of the spillover signal, so that your data processing will be very accurate. And then voltage setting is also very important, because like at very important voltage setting, it should not happen that when you are measuring the cells, if the voltage is very low, they will go out of your measuring range. So when you are doing this, when you are measuring any sample, then setting the voltage also becomes very important. And you can see here that this kind of, in our LSR2, this kind of window comes, and when you this clicks, you can see in the log or A. So what we want to measure, for that you have to click. Suppose that if you want to measure fit C only, then you have to click only fit C, and others you will not click. But when you don't know that when you don't know about that your the sample is in which will show, give the emission in which range, or it gets excited in which range, then you have to probably click most of the channels. So whatever channel it is there, it will automatically click. But if you already know that you are using dye fit C, let's say, then you can click this channel which comes on third, and then it will show, blue laser will hit it, and then your output will come in the fit C channel. So it is based on that. This will give the idea. So immunophenotyping, you can see here, this is one of the application. So let's say if you want to measure some specific expression, let's say you want to measure a stem cell. So a stem cell will have some specific markers on the cell surface. So you will use the antibody against that marker, let's say CD44. So you'll measure CD44 on the surface. So your primary antibody will go and bind on the CD44, and then you will have a secondary antibody which will have a fluorophore tagged, maybe fit C or P or anything. So when you will it will go and bind there, and then after this entire processing of cells, you will run those cells, or if you analyze that cells on flow cytometer, it will show different population of the cells having different level of expression of those that specific dye. So based on your entire WBC population, you can, or if you have taken out from bone marrow stem cell, then you can analyze those cells, that what are different population size. And this is not only one or two markers. People measure, especially when they are doing immunophenotyping or stem cell measurement, they measure more than 4, 5, 5 markers together. And that is why compensation becomes very important in this kind of scenario. And then more than compensation, that the differential analysis also become very important for this kind of analysis or immunophenotypic measurement. You can see that when you are measuring ROS, so what is happening, that when you are treating the cells with any drug, like majority of the anti-cancer drugs, they kill the cancer cells by inducing ROS. So you can see on the left side, you measured, you set by setting the proper voltage, you said that all the cells are coming in this P1 region. And whatever this P1 region, you can see that it is showing two different populations. So one population is there, the one which is not at all showing any ROS, the other population is showing very high ROS. So on the graph, which is on the below graph, you can see here the black histogram. It suggests that when so in this kind of experiments, you need to have one sample where you have not put the DCFDA. DCFDA is a dye which can which gets I mean it's called H2 DCFDA. So when you have, I mean when you treat the, when you stain the cells with H2 DCFDA, it is a what you call lipophilic stain. It goes inside the cell and it gets, I mean it is a diester, so based on esterase activity, its ester will be cut, so it will get trapped inside the cell, and its H2 will get oxidized with reactive oxygen species, and once it gets oxidized, it becomes fluorescent. So you basically measure that fluorescence in FITC, so in FITC channel. So in the black one, there is no staining, so you can see it is on the left side, and when you stained the cells, you can see it is a red histogram in the lower graph. You can see it is coming on the right side. So what it suggests that when you stained, there is an increase in reactive oxygen in the even in the control also. So these are the control cells only. In the black, it is unstained, and the right one, the red one, is the stained one. So the control has this much ROS. So when you treated it with some drug, you can see here H2O2 is generally considered, H2O2 is already a reactive oxygen species generation it causes, and it oxidizes the DCFDA also. So in the top panel, you can see on the here you can see that this is actually microscopy of the DCFDA. So you can see the green color in the left side is left image, it is a control. In the medium one, it is actually treated with H2O2, and the right one actually is having H2O2 as well as an antioxidant. So when you added that, you can see on the middle one, there is huge increase in green color, which is suggestive of there is huge increase in reactive oxygen species generation. But this antioxidant can bring it down. So that is what you measure through microscopy. But if you want to know the quantitative estimation, you need to come here in the down one. You can see this is measured by flow cytometric measurement it is. And the black peak here also you can see 0. That means you have not added anything here. So it is not showing any stain or any much signal. So this is the basic signal you will get in the control cells, which is now unstained. But when you add green color, you can see here the green line, which is suggesting that this is only DCFDA in control cells. So this much difference is there. This much difference is there between unstained cells and stained cells, right? But when you added with some compound, you can see that there is an increase in ROS, right? This is the positive control, means H2O2, which can give the maximum ROS. So your compound is giving this much increase in ROS. With some other concentration, it is showing this much ROS, right? So this is I mean there is an increase. More right shift means more increase of intensity of the DCFDA signal. That means more ROS is formed. So this is how you analyze that your drug is causing any ROS generation or not. So this is about calcium measurement. You can measure the, like many of these drugs, when you treat the cells with these drugs, they cause change in the calcium concentration also. And that can also be measured very easily by, this is a Indo-1 dye, which shows that there is huge increase in calcium signal. You can see on the moment you added inophore. Inophore, what it does basically, it forms a channel. So because of that channel, a lot of calcium comes out, and that calcium that can be easily measured by the dye on flow cytometry. And this is some of our own experiment. Here you can see, like, like radiation, the left side panel 4 images are in the one cell line HeLa, and the right side, these 4 images are in the another cell line, which is called which is MDMB 231. So here, like when we treat the cells with radiation, we use radiation as a treatment. So when you see treated the cells with radiation, this is control cell, normal cell. You can see majority of the calcium came in the perinuclear region. This white arrow, if you can see on the second image from on the top. So you can see here that white arrow is showing that all the green color accumulated in the this opaque region is basically the nucleus. So majority of the green color is coming in the perinuclear region around the nucleus. So it is basically the mitochondria which gets clustered in near the nucleus. So there is one dye which is called Rho2AM. So when you this dye measures specifically the calcium only in the mitochondria. So you can see here in the lower red panel also you can see that majority of the calcium accumulation can be seen in the perinuclear region near the what you call nucleus in the microscope. So the lower histograms you can see here, this is bar diagrams. They are measured by flow cytometry. So this is actual quantitative estimation you can see in the HeLa and MD-MB 231 cells. The upper graph shows flow 3AM, which measures non-specific the calcium in all over the cells, but the lower one talks about Rho2AM. So you can see here this gives a clear difference that whatever calcium came out during this treatment with radiation, whatever calcium came out, that got predominantly accumulated into the mitochondria, right, in case of radiation. So you can see here the difference is much less in the top bar diagram. If you can see, the difference is top bar diagram flow 3AM showing the difference is much less between control and radiation. But here you can see the difference is very high between the control and radiation. So whatever my calcium came out, that had gone predominantly into the mitochondria. This is what this data suggests. So this information you cannot get from the microscopy. You will get this information from flow cytometry only. And you know, like once calcium accumulation happens into the mitochondria, it will cause the cell death. So this is what basically is this is basically major thing which tells that you can find out the mechanism of action of this drug, whatever drug you are using, or radiation, like let's say we use radiation. So we come to know that what is the mechanism of action of this drug, how this drug is causing death into that specific cell line or other cell lines which you are using. So this is again about apoptosis and necrosis. So here again we used radiation as a treatment. So you can see on the left side, this is control. And in control, you can see that only 8.3% cells are coming in the. So on the y-axis, you will see the PE. PE means propydium iodide. So propydium iodide goes only into the dead cells. It doesn't go into the live cells. So so very high cells moving on y-axis will show that the cells become necrotic or late apoptotic. So these are the things on the right panel, the panel which is on the high on the y-axis and high on the low axis, where you can see 8.3%. This window will suggest that these are the late apoptotic cells. And the panel on the x-axis towards the right, where you can see 9.5%. This panel shows Q4. This talks about this shows that it is the early apoptotic cells. Majority of the cells which will show early apoptosis, they will show only actually phosphatidylserine. So this annexin 5, which is tagged with FTSE or any green color dye, that will measure the early apoptotic cells. the cells coming in the Q2 panel, this panel, that will have high, annexin 5 measurement also, labeling also, and high PI labeling also. So those cells will be late apoptotic cells. Any cells which are coming in this panel, which is Q1, they will be actually the necrotic cells, and all the normal cells will come mainly into this portion, Q3. So this is 79.4%. These are normal cells. These are cells with some stress, due to which you can see some population is showing little bit change into the annexin 5 staining and all. So in IR, when we given the stress, radiation stress, you can see majority of the cells are in this panel, 36.7% cells, more than normal cells, 28.2%. They are in this this panel, this quadrant, which is showing that majority of the cells are dead, and some are early apoptotic. 20% cells are early apoptotic, and 15.1% cells are necrotic. So radiation is causing almost 70% cells are in different form of death. So when we have given the drug, you can see here, this is a drug with the radiation. This is only drug. So drug is not causing much difference than the control cells. But when we have given drug and then given the radiation, you can see the level has gone significantly down. As compared to 36.7%, it is just 22.3%. And this is less than 20%. This is coming down to 10% only. 10% only. And this 21, this necrotic cells are relatively higher, but majority of the cells has come down. And as compared to 30% cells, you are getting almost roughly 50% cells here. So 20% cells you are able to save. So this is what this drug is doing. You can save 20% more cells in this by treating with this drug. So you can easily find out that your drug is, whether your drug is working or not, when you have treated it with the radiation. So suppose that if you want to develop any drug which is actually can work good against radiation, can be a good radio protector, then you need to know that whether that drug can reduce the oxidative stress or not, whether that drug can reduce the apoptosis or necrosis or cell death or not. So these parameters measurement will help in deciding that. So this is this is cell cycle. So cell cycle measurement, I think it will not, it will be little difficult to understand here. You can see here in cell cycle, these are again in those two cell lines. Whatever you are seeing a large peak here, that shows the G1 content. Majority of the cells are in G1 population, 74% cells, and only 5% cells are there in S phase. That means DNA synthesis is going on between this portion. So this is G1 at 200, and then at 400 the DNA count will be double. So G2M cells will have 400, will come at 400, and G1 cell will come at 200, because like in G2 and M phase, this DNA content become just double. So it will, from 200 it will become 400. So any cells between this G2 and G1 is the cell between in the S phase. So many of the drugs, they disturb this S phase also. So when radiation is given, it because to happen the DNA repair, the cell cannot divide before it repairs all the cells. So that is why you can see that the G2M population has gone high from 11% to it has gone to 28%. Right? So that is why you are seeing a bigger peak here. So whether your drug is causing this kind of G2M block or not, that you can analyze by doing cell cycle on flow cytometer. So this will give a very good evidence, and this is also telling that whatever block happened, that happened only at 24 hours. Later, when you analyze the same treated cells at 48 hours, you find that this is almost like a control cells only. Here it was 11%, here it is 12%. So that means all the cells which were blocked, that is gone. The same observation you can see in another cell line also. So here there are certain more evidences and for the application. So this is again from our lab. We analyze certain drug. The good thing with this drug that this doxorubicin drug itself is a fluorescent drug. So there was some nanoparticle was designed. This is the nanoparticle PBLG23BHYA. Based on the PEG, this was made, and then this doxorubicin was actually packed in this nanoparticle. So and these specific nanoparticles, they can target the CD44 positive cells. So you can see here we measured that MCF7 cells has high level of CD44 expression. So this upper graph also, this measurement was done by immunophenotyping. We used antibody against CD44 and tagged them, and then see it can be done in two step also, like when you have primary antibody and then secondary conjugated antibody with fluorophore, or many a times what happens that you get the primary antibody itself conjugated with the fluorophore. So you do that in one step. So here we measured that what is the level in two different cell lines, what is the level of CD44, and we found that MCF7 cells have very high level of CD44, and the accumulation of the drug also you can see is very high as compared to doxorubicin. The accumulation of the cell dye also is very high in these cells. But you can see a very unique thing in the doxorubicin. You will see that majority of the doxorubicin is accumulated into the nucleus. And you know, doxorubicin is basically DNA damaging agent. So, but what we found that in these nanoparticle, the cell death was very high as compared to doxorubicin treated cells. So, but this drug is not going to the nucleus. You can see here in the lower panel, the drug is not in the nucleus. It is in the outside the nucleus, in the accumulated in the nanoparticles. So that is why we thought that probably it is not causing cell death by causing DNA damage. Probably it is causing cell death by some other mechanism. So you can study actually the drug uptake also by flow cytometry. You can see here at 3 hour, 6 hour and 24 hour, this was drug uptake was measured using in different cell lines using this flow cytometry. So here you can see here that the drug uptake was measured. You can see that the drug uptake is going high up to 20 hours. And then the drug release is probably happening. This is the free dox you can see here, and then different kind nano particles which we made. Which one is showing the best uptake of the drug? Which nanoparticle shows the best uptake of the drug can also be analyzed using flow cytometry. So this is what we have shown here. Here again, this method methotrexate was entrapped in some new nanoparticles, and you can see here as compared to microscopy how the death is happening. That can be seen and analyzed by the microscopy. By flow cytometer. This is another parameter where we measured that how to measure the mitochondrial membrane potential. You know, for measuring the cell death, mitochondrial membrane potential is very important. And what we were doing, I mean, in these experiments, we treated the cells with mitochondrial uncoupler. And uncoupler, what it does, it brings down the mitochondrial membrane potential. So you can see here that on the left side there is a control. So all these orange colors dots which you are seeing, they are the mitochondria with high membrane potential. And then the green color mitochondria are basically only mitochondria with low membrane potential. So in control, majority of the cells are having mixed kind of high or low mitochondrial membrane potential. These are different treated with different mitochondrial uncouplers, different concentration. So this is the first one is DNP treated. Then at 10 micromolar, you can see that very mild uncoupling is happening. In BHT 1 micromolar, again a strong mitochondrial uncoupling is happening. At BAM 15 1 micromolar, these are different drugs, mitochondrial uncoupler, and they are causing different level of mitochondrial uncoupling. At high concentration of all the drugs, you can see there is hardly any orange color. So that means there is no there is hardly any mitochondria which is having high mitochondrial membrane potential. Majority of the mitochondria is showing green color. That means the mitochondrial membrane potential is very low. But you need to comment exactly how much low it has gone, how many fold it has gone down. So that is what we measured here. You can see on the left side all the images which you can see on the top panel. You can see they have mild mitochondrial uncoupling. So we have to our target was to do mild mitochondrial uncoupling. Our target was not to completely uncouple the mitochondrial membrane potential, reduce the mitochondrial membrane potential. So that is why we had to standardize the concentration, at what concentration it is doing mild mitochondrial uncoupling. And for that, microscopy was not sufficient. We had to go to do the flow cytometry to analyze this, that at what concentration the mild mitochondrial uncoupling can be obtained, which is actually protect the cells against radiation induced damage. So for that, so this becomes very important, the concentration of the drug, to find out the concentration of drug which can be protective, because at high concentration, the drug starts killing the cells. The 10 time, when you increase the 10 time concentration, it starts killing the cells, because extremely low mitochondrial membrane potential will initiate the cell death in the cells. So that is why it is very important. So as I said in the previous one, that how to analyze the mechanism of action, what we did, you can see in the top left side top image, majority of the doxorubicin is coming in the nucleus. So here the cell death must be happening because of the DNA damage. But in the lower one, where it is trapped into the nanoparticle, you can see a hollow here, a big hollow here, that is the nucleus. This big hollow is a nucleus. So drug is not gone into the nucleus. It is all around the nucleus, outside the nucleus. So that means there must be some other mechanism. So we measured that reactive oxygen species generation, because doxorubicin kill the cells by ROS generation also. And we found, you can see here, the black color polydox, that is the nanoparticle, the ROS generation is very high in this. So that means it is basically in these cells, in these nanoparticles, the cell death is high because of high ROS generation. At 6R, the level is at 6R, the level is low. In the polydox, at 24R, the level is even further high. And we showed that the drug uptake is maximum at 24R, between 20 to 24R. So that is why this is probably happening a very strong. And you know, doxorubicin, a lot of cancer cells show resistance towards this kind of drug, also because they efflux out. So when you packed this in this nanoparticle, the cell was not able to efflux it out, and most of the cell death was happening at that point of time. So this flow cytometry can be very well utilized in understanding the mechanism of action also. So without understanding and processing the cells, you can separate the cells also based on their intrinsic or extrinsic parameters. So suppose that if you want to, in any given population, the same cell, if you want to know that some cells express CD44, but other cells are in the same cells, some cells are not expressing the CD44. Or if you want to segregate the cells based on, like some cells which are showing high ROS, some cells are not showing low ROS. So by doing the staining, you can segregate them. That is called cell sorting. So in normal flow cytometer, this facility does doesn't come. If you are buying a high-end flow cytometer along with sorter, then you can get this this facility also, where the stained cells will be given. With the staining, you will give a positive charge to that, and unstained cells will go into with the negative charge, and they will get collected into two different vials, and then you can take out those cells and you can culture them. So you can culture the cells with different see, majority of the cell culture when you take, they are actually the heterogeneous population. So if you want to, let's say if you want to segregate the cells of G1 with the cells of G2, so that's how also you can, based on this staining, different markers which express only in G1 and different markers which express only in G2, you can stain them and you can segregate those cells, and you can propagate the homogeneous cells with different properties, with different characteristics, etc. So the cell sorting can also be used for that kind of application. So thank you very much. If there is any question, then I can take the questions.
Rupali madam, can I take a moment before the question answer round?
Yes, sir.
Okay. Thank you very much, sir. What the best part of the Anand sir is that sir is, along with the English language, sir is having a good command of the Hindi language, and in his organization, sir is always used to work in the the Hindi language for the promotion of this Rashtriya Bhasha. So I would like, if the Anand sir is comfortable, so I would like to conduct this question answer round in the Hindi. Sir, are you comfortable with this thing?
हाँ, बिल्कुल बिल्कुल.
Thank you very much, sir. सर, पहला क्वेश्चन तो मैं ही आपसे पूछना चाहता हूँ. जैसा कि आपने बताया कि जो फ्लो साइटोमीटर,
Nitin, your voice is not coming.
नितिन सर, आपका माइक म्यूट हो गया है.
सर मेरी आवाज आ रही है आपको?
अब आ रही है.
हाँ नितिन, आ रही है.
हाँ सर, जैसा आपने बताया कि फ्लो साइटोमीटर से हम किसी भी सेल का ग्रेनुलेशन चेक कर सकते हैं, तो मेरा एक ही नॉर्मल डाउट है, और नॉर्मली जो फार्मेसी फैकल्टी हैं उनका भी ये डाउट हो सकता है कि सपोज हम लोग यदि कभी कोई ट्रीटमेंट करते हैं किसी सेल लाइंस के ऊपर, और क्या ये ट्रीटमेंट हमारे ग्रेनुलेशन को इफेक्ट करता है? मींस यदि मैं ये कहता हूँ कि दिस ट्रीटमेंट, ये जो ट्रीटमेंट है ये आरओएस लेवल को अप रेगुलेट कर रहा है या आरओएस लेवल को कम कर रहा है, या कोई पर्टिकुलर प्रोटीन को एक्सप्रेशन को बढ़ा रहा है या कम कर रहा है, तो क्या ये ग्रेनुलेशन उस टर्म से कोरिलेट हम कर सकते हैं?
हाँ डेफिनेटली नितिन, ये जो जैसे ढेर सारे ट्रीटमेंट के बाद सेल का साइज चेंज होना और ग्रेनुलेशन चेंज होना, ये बहुत कॉमन चीज है. जैसे हमारी लैब में ही एक सेल है, उसको हमने ट्रीट किया, उसका मेटाबॉलिज्म अल्टर किया, तो मेटाबॉलिज्म अल्टर करने के बाद उसकी ग्रैनुलैरिटी बहुत बढ़ गई, और वो क्लियरली दिखता है. जैसे अगर हम जो पेरेंट सेल है उसको मेजर करें तो उसका स्कैटर बहुत वाइड आता है, लेकिन जब हम इस सेल को मेजर करते हैं तो वो सारी सेल जो है वो बहुत ग्रैनुलर हो गई है, और बहुत उसका स्कैटर जो आता है वो बहुत क्लोज में आता है, और उसकी ग्रैनुलैरिटी जो है वो पेरेंट सेल से बढ़ी हुई है. So this is a common observation. You treatment के बाद you will get this kind of changes in the cells.
Okay sir. अच्छा, क्या सर हम इसमें अपना सेल हमारा लाइव है या डेड है, ये चीज भी हम ग्रेनुलेशन के बेस पे डिस्टिंग्विश कर सकते हैं?
जैसा कि मैंने बताया कि जो डेब्रिस सेल्स होता है उसकी ग्रैनुलैरिटी बहुत ही लो हो जाती है. जो सेल डेब्रिस होता है, डेड हो जाने के बाद उसकी ग्रैनुलैरिटी जो होती है वो बहुत घट जाती है. तो वो दैट विल कम इन द क्लोज, आई मीन कॉर्नर में, जो जहां से एक्स और वाई एक्सेस शुरू हो रही है लेफ्ट कॉर्नर में, वो ज्यादातर वो सेल्स उसी एरिया में आएंगी, डॉट्स जो होंगे.
धन्यवाद सर. रूपाली मैम, क्या हम और आगे सवाल ले सकते हैं?
Sir, there is one question from Ms. Nidhi. I want to ask that a good anti-cancer agent.
विपिन सर प्लीज मैं रिक्वेस्ट कर रहा हूँ कि हम ये जो क्वेश्चन आंसर राउंड है उसको पूरी तरह से हिंदी में रखेंगे, जस्ट टू पे द ट्रिब्यूट टू आवर मदर लैंग्वेज।
अच्छा, ये एक्चुअली इन्होंने इंग्लिश में टाइप किया है कि
सर आप क्वेश्चन हिंदी में इंग्लिश में बता दीजिए सर, आप उसका हिंदी में यदि आंसर दे सकते हैं तो प्लीज।
बिल्कुल बिल्कुल बिल्कुल।
क्वेश्चन है कि a good anti-cancer agent should show which type of apoptosis, early or late?
सी ये अर्ली और लेट फेजेस हैं उसकी, मतलब जैसे जब ये डिपेंड करता है, जैसे अगर आप ड्रग देने के 12 या 24 घंटे बाद ही मेजर कर लिए तो आपको कुछ सेल्स मिलेंगी जो अर्ली एपोटोसिस में मिलेंगी, कुछ लेट एपोटोसिस में मिलेंगी। और जब क्या नाम है उसी सेल पॉपुलेशन को 36 घंटे के ट्रीटमेंट के बाद आप मेजर करोगे तो ज्यादातर सेल्स जो है वो अर्ली एपोटोसिस से लेट एपोटोसिस में मूव कर चुकी होंगी, और जो कुछ कंट्रोल सेल्स होंगी वो हो सकता है कि कंट्रोल से फिर वो अर्ली एपोटोसिस में मूव कर गई हो। सो ये फेज है, ये एक मतलब सेल डेथ का फेज है। जैसे आपने 100 सेल्स को ट्रीट किया तो आपको हो सकता है कि बहुत अर्ली आपने मेजर कर लिया तो मेजॉरिटी ऑफ द सेल्स जैसे 20 अभी भी नॉर्मल हैं और 50-60 जो है वो अर्ली एपोटोसिस में मिलेंगी और 20-30 लेट एपोटोसिस में मिलेंगी। लेकिन जैसे जैसे ये टाइम बढ़ाते जाएंगे तो मेजॉरिटी ऑफ द सेल्स विल शिफ्ट टुवर्ड्स लेट एपोटोसिस और डेब्रिस।
हेलो सर, संदीप सेन सर हैज रेज्ड हिज हैंड। संदीप सर, आपको कोई क्वेश्चन पूछना है, आप पूछ सकते हैं।
अच्छा सर, मेरा आपसे क्वेश्चन पूछना था। जैसे सर हम लोग जैसे ये जो है आपका इस प्रोसेस में हम लोग जनरली एंटी कैंसर ड्रग्स के केस में बहुत हेल्प मिलेगा, लेकिन सपोज पकड़ लीजिए कोई एंटी डायबिटिक ड्रग हुआ या कोई कार्डियोवास्कुलर ड्रग हुआ, उस केस में अगर सपोज हमारा टारगेट तो न्यूक्लियर रिसेप्टर के ऊपर काम नहीं करता, हमारा रिसेप्टर दूसरे किस्म का रिसेप्टर होता है, तो उस केस में हम लोग स्टडी कर पाएंगे इस केस में?
हाँ, कोई भी स्टडी, जैसे सपोज दैट आपका जो ड्रग है वो ग्लूकोज अपटेक बढ़ा रहा है या नहीं बढ़ा रहा है, आप सेल्स में मेजर करना चाहते हैं तो आप ग्लूकोज के फ्लोरेसेंट एनालॉग आते हैं, आप उनको कंट्रोल सेल में ग्लूकोज डाल दीजिए। जो डायबिटिक सेल है मान लीजिए उसका अपटेक नहीं हो रहा है, ग्लूकोज अपटेक उसका कम है, तो कंट्रोल सेल में मेजर कर लीजिए और अपना ड्रग ट्रीटमेंट के बाद वो जो फ्लोरेसेंट एनालॉग है ग्लूकोज का उसको 2-NBDG बोलते हैं, वो आप उसको डाल के अपने ड्रग के साथ और 2-NBDG से स्टेन कर लीजिए। तो 20-30 मिनट का स्टेनिंग होता है, उससे आपको पता चलेगा कि अगर आपका ड्रग ग्लूकोज अपटेक को बढ़ा रहा है तो जाहिर सी बात है जो आपका ड्रग ट्रीटेड सेल है उसमें फ्लोरेसेंस इंटेंसिटी ज्यादा मिलेगा आपको एज कंपेयर टू विदाउट ड्रग ट्रीटेड ग्रुप।
इसमें और एक चीज मुझे क्लियर करना था, जैसे इस केस के अंदर जैसे हम लोग जब कोई ड्रग्स जनरली कोई एनिमल मॉडल यूज करते हैं ना, तो एनिमल मॉडल पे सपोज पकड़ लीजिए हम लोग कोई रेट जैसे कंट्रोल ग्रुप में है, एक रेट जो है वो ट्रीटमेंट में है, तो उस केस में जैसे पकड़ लीजिए हम लोग रेट से जैसे इस केस में हम लोग रेट का जो ऑर्गन्स है उनको लेकर डायरेक्ट स्टडी कर सकते हैं या हमें सेल लाइन बनाना पड़ेगा?
नहीं, आप ऐसे भी ऑर्गन निकाल के उसको टिश्यू को प्रोसेस करके उसको सेलुलर फॉर्म में ले आ सकते हैं। उसमें क्या होता है, काफी कनेक्टिव टिश्यू वगैरह सब आपको हटाना पड़ता है, फिल्टर करके आपको सेलुलर फ्रैक्शन में ले आना है, उसके बाद आप स्टडी कर सकते हैं।
अच्छा
जी,
धन्यवाद सर।
वंदना मैडम, आप क्वेश्चन पूछ सकते हैं अपना।
या श्योर, थैंक यू सो मच सर, आपका प्रेजेंटेशन बहुत अच्छा लगा और बहुत कुछ समझ में भी आया। आई मीन फ्लो साइटोमेट्री इतने डिटेल में तो शायद मैंने पहली बार पढ़ा। सर मेरा एक क्वेश्चन है, आई मीन हमको अगर फ्लो साइटोमेट्री किसी फ्रोजन स्टेम सेल पे करना हो तो हम कैसे करेंगे? आई मीन उसको हमें 1 नाइट और 24 आवर्स पहले उसको कल्चर करना पड़ेगा फिर ही हम काम कर सकते हैं, क्या प्रोसीजर रहेगा सर?
जैसे अगर उसको कल्चर करने का रिक्वायरमेंट नहीं है आपको, आप सैंपल जैसे फ्रोजन सैंपल्स होते हैं उसको भी अगर आपको मेजर करना है तो आप उसको पहले थॉ कर लीजिए मतलब जो भी प्रोसीजर है थॉ करने का, उससे थॉ कर लीजिए ताकि सेलुलर डैमेज ना हो। एक्चुअली फ्रीज थॉ में यही प्रॉब्लम होगा कि उसमें सेलुलर डैमेज हो जाएगा तो नेक्रोटिक हो जाएंगी, बहुत सी सेल्स डिब्रिस में आ जाएंगी। तो आप उसको प्रॉपर तरीके से थॉ कीजिए और उसके बाद उसका जो भी प्रॉपर्टी स्टडी करना है आपको, सपोज दैट आप फ्लोरेसेंस उसमें आरओएस मेजर करना चाहते हैं या स्टेम सेल है तो कोई मार्कर मेजर करना चाहते हैं, तो वो उसको स्टेनिंग करके, मतलब उसको जैसे इम्यूनोफिनोटाइपिंग करनी है तो आपको इम्यूनोफिनोटाइपिंग के लिए प्राइमरी सेकेंडरी के साथ स्टेनिंग करके और जब ब्लॉकिंग करके जो भी मतलब स्टेप्स हैं फॉलो करके यू कैन मेजर इट, आई मीन थॉ करके ही। अगर आप कल्चर करके उसको फिर स्टडी करना चाहते हैं तो फिर आप उसको कल्चर में ले जाना होगा।
ओके, अच्छा, यानी कोई मैंडेट नहीं है, इट विल मतलब वो हमारा प्रोसीजर और रिक्वायरमेंट के हिसाब से हम थोड़ा बहुत मॉडिफाई कर सकते हैं।
हाँ, आपको सैंपल में डायरेक्टली अगर करना है तो आप डायरेक्टली भी कर सकते हैं फ्लो।
ओके ओके, थैंक यू सर।
ओके,
अंजना मैडम आप कुछ पूछना चाहते थे? अंजना मैडम? हाँ मैडम, आप अनम्यूट कर लीजिए अपने को।
Good afternoon, sir.
गुड आफ्टरनून।
Sir, the session was very informative and जो है मेरा यही क्वेश्चन था जो वंदना मैम से रिलेटेड जो आपने क्वेरी बताया है उससे क्लियर हो गया मेरा सर। थैंक यू सर।
ओके, थैंक यू।
और किसी पार्टिसिपेंट का कोई क्वेश्चन है तो आप अपना हाथ रेज कर सकते हैं, तो हम आपको अनम्यूट कर देंगे। रूपाली मैडम, मेरे ख्याल से अब कोई क्वेश्चन नहीं है, तो इट्स ओवर टू यू नाउ।
Sir, I would like to thank you from the bottom of my heart for taking time from your busy schedule to be the speaker in this program. And your knowledge helped magnify the use of flow cytometry in the best possible way. Thanks a lot, sir. Thanks a lot.
Thank you. Thank you, organizers.
थैंक यू अनंत सर।
Bye, नमस्कार।
Nitin sir, can we end the meeting now?
Yes, sir.
Okay, so tomorrow again for the last day of the session we will be meeting at 9:30, and tomorrow we have 2 hands-on sessions. So tomorrow at 9:30 we will be meeting. Till then, goodbye and good evening to all.
Thank you, Vipin sir.
Thank you. Exam will be tomorrow only.